Hello, everyone. My name is Chaoyang Yan, and I am a second-year PhD student at Nankai University in China. Today, I am honored to present our work at ISMB 2024 on a phenotype-informed weakly supervised model for HER2 status prediction from pathological images, which we have termed PhiHER2. First, I would like to extend my gratitude to my co-authors, Jialiang Sun, Yiming Guan, Juxing Feng, Hong Liu, and my advisor, Jian Liu, whose contributions were invaluable to this research. Now let me introduce the research problem. HER2, human epidermal growth factor receptor 2, is an important protein involved in the regulation of cell growth and division. It is amplified or overexpressed in approximately 15 to 20% of breast cancer (BC) cases. This subtype of breast cancer tends to be more aggressive and is associated with a poorer prognosis. Importantly, HER2 overexpressing breast cancer is responsive to anti-HER2 therapies, and there are several therapeutic agents available for clinical use. These include trastuzumab, pertuzumab, tyrosine kinase inhibitors, TKIs such as lapatinib and tucatinib, and antibody-drug conjugates, ADCs, like TDM1 and TDXD. HER2-targeted drugs significantly improve clinical outcomes in patients with HER2-positive breast cancer and have reshaped the diagnosis and treatment landscape of breast cancer. Thus, the precise determination of HER2 status is crucial as it helps clinicians develop individualized treatment plans and improve treatment outcomes. However, in clinical practice, several challenges hinder the efficient and accurate determination of HER2 status. The IHC diagnostic process is time-consuming and expensive. Experiments in sample handling are complicated and often uncontrolled. HER2 expression levels from IHC are qualitative and semi-quantitative. The process is influenced by subjective, professional, and other uncertain factors, leading to inconsistency. Therefore, an efficient, accurate, and fully quantitative method is highly required for HER2 status detection in breast cancer. Histopathology serves as the gold standard for clinical diagnosis. Hematoxylin and eosin-stained pathological slides provide morphological information accompanied by spatial organization. Nowadays, with the advent of artificial intelligence, computational pathology has experienced significant advancements through digital whole-slide image analysis. It has shown remarkable progress in various tasks, including nuclei and tissue identification, tumor detection, cancer grading, subtyping, survival prediction, treatment response prediction, and prediction of molecular targets, among others. Computational analysis of pathological images holds the potential to unveil morphological patterns associated with HER2 molecular targets and achieve precise status prediction. However, pathological images are characterized by high-resolution, multiscale attributes, often reaching billions of pixels in size. This presents a significant challenge for standard CNNs in accurately identifying regions of interest and aggregating features. The current paradigm utilizes a multi-instance learning framework, a weakly supervised learning method that encompasses whole-slide image processing, feature extraction, feature aggregation, and HER2 status prediction classifier head. In preprocessing WSIs, the primary objective is to eliminate non-tissue background areas and segment WSIs into smaller patches suitable for MEL. Feature extraction plays a crucial role in this process, serving as a fundamental transformation function. Therefore, our research problem can be denoted by this formula. Recently, a review has summarized the evidence concerning the biological and clinical significance of HER2 heterogeneity in solid tumors of breast cancer undergoing HER2-targeted therapy. HER2 heterogeneity is categorized into intratumoral, intertumor spatial level, and temporal level variations. Intratumoral spatial heterogeneity has been associated with poorer therapeutic responses to HER2 therapy. Given these findings, an intriguing question arises. Can this HER2 heterogeneity within tumor tissues be discerned from the histomorphologic level? We discover histomorphologic phenotypes from histopathological tiles in WSIs. Considering the number of instances, we have developed a two-stage hierarchical prototype clustering module using the unsupervised affinity propagation AP clustering algorithm. In the first stage, local AP clustering is iteratively conducted on each slide with instance features, resulting in cluster centers. These centers from individual slides are then combined. In the second stage, global AP clustering is applied across all WSIs to obtain final cluster centers. This method ensures robustness to initial values and determines the optimal number of clusters for diverse instances in WSIs, capturing representative patterns that reveal tissue heterogeneity. We introduce this hierarchical AP clustering as function phi into the feature aggregation process for guidance. Leveraging this characteristic enhances HER2 status prediction. The overall flowchart of our phenotype-informed framework is illustrated with the hierarchical prototype clustering module capturing representative morphological patterns in WSIs. We employ a cross-attention module for feature aggregation to better understand tissue heterogeneity through phenotype embeddings. Based on phenotype-based slide-level representations, various classifier heads are explored for HER2 status prediction, including mean-based, attention score-based, and transformer-based heads. We validated our model on two datasets, the HER2 dataset and the Yale cohort. Key experimental details include 1. Feature extraction using REAT-CCL for instance-level embeddings resulting in 2048 dimensional vectors. 2. Optimization using cross-entropy loss, ELFBI, as the overall objective function to train our PhiHER2 model. For further comprehensive details, please refer to our paper. To verify the effectiveness of our PhiHER2 model, we chose several state-of-the-art weakly supervised methods, ABMEL, CLAM, PMEL, and Transformer for comparison. The results demonstrate superior performance of our PhiHER2 in accurately identifying HER2 status from BCWSIs. The baseline ABMEL and the CLAM method, enhanced with instance attention scores, yielded acceptable but unremarkable results for the specific molecular HER2 status prediction task. The PML cosine and PML Euclidean methods outperformed CLAM and ABMEL on both datasets. This points out the advantages of phenotype-informed architecture. The evaluation analysis involved ROC and PR curves, along with average AUC and AUP RC values, are also presented on the left. It achieved the best AUC values of 0.795 and 0.890 on the HER2 dataset and the Yale cohort, respectively. We further categorized the comparative models into two groups, phenotype-guided and non-phenotype-guided. The phenotype-guided group consistently provides better performance, confirming the superiority of the concept of phenotype guidance. Given that the HER2 dataset was from a competition, we benchmarked our results against the top entries on the public leaderboard. Our method outperformed the first-place entry, achieving a 2.6% improvement in terms of the AUC metric. Our method obtained a notable improvement of 8.5% over the first-place entry. We investigated the distribution of predictions on different HER2 status groups. The predicted probabilities for HER2 positive on the HER2 test cases were examined. It could be observed that our method exhibits the widest gap between the median values of the prediction probabilities across different HER2 status groups. These findings suggest that our PhiHER2 has the most significant discrimination between HER2 positive and negative cases. To validate the effectiveness of our proposed hierarchical prototype clustering structure and the cross-attention module, we adopted 4 distinct experimental configurations for comparison. The architectures of experimental configurations are as follows. I, cluster PT, the model with a cross-attention module and phenotype embeddings derived from hierarchical prototype clustering structure to provide guidance. The T, RAN PT, fixed random vectors served as phenotype embeddings in the cross-attention module for direct inference. Random values were sampled from a uniform distribution with dimensions and quantity identical to those of the cluster phenotype embeddings. E, initial PT, random vectors initialized with a uniform distribution served as phenotype embeddings. These vectors could be iteratively optimized during the training process. Non-PT, the model with a cross-attention module but no phenotype guidance. In this configuration, the cross-attention module reverts to a self-attention mechanism. We also included the ABMEL method as baseline for comparison. Cluster PT consistently outperformed the baseline ABMEL and non-PT on both the HER2 dataset and the Yale cohort. Cluster PT obtained an average AUC value of 0.893, marking a significant improvement of 6.7% over non-PT and 7.3% over the baseline on the Yale cohort. Cluster PT approach achieved superior performance compared to both RAN PT and initial PT, with respective AUC improvements of 10.1% and 1.0% on the HER2 dataset. We also compared the performance of our PhiHER2 model when coupled with different classifier heads. covering those based on mean operation, attention scores, and transformer strategies. Our PhiHER2 remains relatively robust with respect to different classifier head strategies, and these models outperform the baseline ADMO method across both the HERO-A dataset and the Yale cohort. We thus obtain the WSI level representation vector before the classifier head and visualize them by embedding heat maps. Our phenotype-informed aggregated features reveal a significant distinction between the HER2 positive and negative groups, displaying greater embedding consistency than those of PMIL and non-phenotype guided methods. In this section, we provided the tissue phenotypes corresponding to the prototype embeddings derived from the hierarchical prototype clustering module. We can find that the clustering module captures typical phenotypes within WSIs. Expert pathologists characterize these regional tissue phenotypes as dense tumor cellularity, a discrete distribution of tumor cells, abundant stromal fibrosis, the presence of tumor-infiltrating lymphocytes, and a white background. We employed a pre-trained hovernet for cell segmentation and classification on these regional tissue phenotypes. This allowed us to obtain the cellular spatial composition of neoplastic, inflammatory, connective, dead, and non-neoplastic epithelial cell types. Some phenotypes showed a high abundance of locally aggregated inflammatory cells and a higher percentage of tumor cells, while others exhibited loosely distributed stromal cells interconnected with tumor cells. These observations align closely with those made by expert pathologists. This analysis reveals distinct cellular spatial compositions and morphological patterns in pathological images, potentially offering explainable insights into the spatial morphological heterogeneity of breast cancer. Additionally, we conducted a 2D density plot analysis on the prototype embeddings with model prediction scores to compare HER2 positive cases, denoted in purple, with HER2 negative cases, blue. A clear separation is evident between the two groups. Recognizing the importance of interpretability in clinical decisions, we implemented an overlapping heat map visualization method to assess the importance of regions and interpret the morphological patterns associated with the molecular status of HER2. Fine-grained heat maps overlaid on WSIs from the Yale cohort are visualized here. The WSI heat maps demonstrate a high degree of consistency with expert annotations, denoted in green, of regions of interest ROIs in HER2 positive and negative cases. Our PhiHER2 model effectively identifies and weights ROIs related to HER2 status using slide-level labels only. Most of the high contribution areas are located within tumor tissue, with the representative patches having the highest probabilities predominantly derived from tumor areas. This underscores the interpretability of which tissue patterns are most significant for making WSI-level HER2 status predictions. Overall, we developed a phenotype-informed weakly supervised computational model for HER2 diagnosis from histopathological slides. We discovered heterogeneous phenotypes and cellular spatial composition from the histomorphological perspective. We explored the morphological patterns associated with the HER2 status at the spatial level. We provided new insights into the correlation between molecular biomarkers and pathological tumor tissue morphology. Okay. Thank you. Thank you for your attention today. Have a good night.